So hello everyone. I welcome all of you for yet another Fusion Talk. On behalf of Pre-Science in Silico, I welcome all of you to this talk. I'll speak few words about Pre-Science in Silico. At Pre-Science in Silico, we team up with both industry and academia to tackle challenges in drug discovery and material science space. The idea for this Fusion Talk series, it has came from our wish to get experts from different fields to share their valuable thoughts. And we are very proud of to be one of the first Indian companies to create our own drug discovery software with advanced AI features. Prescience in Silico was founded by academicians who wanted to make AI powered discoveries in drugs and materials accessible to everyone to aiming to redefine the landscape of scientific knowledge. And it is our privilege to have Professor Praveen Kumar Vemula with us today, who will be delivering talk. Few words about Professor Vemula. Professor Vemula is a professor and Dean of Research at the Institute for Stem Cell Science and Regenerative Medicine. It's called as inStem in Bangalore. He has done his PhD from IISc, followed by his postdoc at New York City College, followed by another postdoc at Harvard Medical School. He has his expertise in developing chemical technologies for medical applications, and his work spans in the field of biomaterials, drug discovery, drug delivery, and medical devices. Professor Vemula has published more than 95 peer-reviewed papers and given more than 300 national and international invited lectures, including three TEDx talks. And he has more than 30 issued or pending national or international patents, which have been licensed to multiple biotech companies. Based on his technologies, he has formed around 8 startup companies, and they have more than 25 products developed based on his technologies and which are in market worldwide. Along with this, he has his work has been recognized by Institute of Chemical Technology, RSC, that is Royal Society of Chemistry Innovation Award. And he has also received National Biotechnology Innovation Award 2023, DBT Product Process Technology Development and Commercialization Award in 2020, and GYTI Srishti Birak Award in 2019. So with this, I would hand over stage to Professor Vemula, and we'll be looking forward to the interesting talk from him.
Thank you so much, Anil, for this kind words, and also thanks for inviting me to share some of our work with this audience. And today, particularly, I'm going to talk about how a chemical biology approaches could be used for solving unmet clinical need. However, before I start the talk, as a protocol, I need to disclose the conflict of interest. These are the companies I formed based on the technologies what we developed in my lab. I'm co-founder of these companies. I'm director, non-executive director in this, and then I also own the equity in these companies, and I get the financial benefits. And the work what I'm going to talk about it, the part of some of these companies. With that, my lab at inStem, we primarily focused on clinical translational research, where two major programs we develop in our laboratory, like the biomaterials and chemical biology programs. Whereas in the biomaterials, it's like primarily we design next generation biomaterials to improve the efficacy of the existing drugs by delivering them very innovative manner. But whereas the chemical biology program, it's a classical new drug discovery program. Okay, and as name suggests, biomaterials could be use them as a therapeutic technologies to where you can use them to treat the diseases, or you can use them to prevent the diseases to occur like prophylactic technologies, right? And in the drug discovery program, primarily we use there are two major approaches. Just a second. Okay. Yeah. So can I take out the footing? Just a second. I'm just trying to see how we can take this off.
Sure, no problem.
Yeah, I think this is far better. So again, like we use like both small molecular based new chemical entities we develop as a drug, and also the novel biologics also could be developed as a drug. So all our work comes in part of one of this category. And today I'm going to talk some of the examples from these approaches. To begin with, like, we have been like focusing on developing technology for the therapeutic approach. Particularly, like we were interested in developing one of the platform technology where because like if you see the inflammation is the inherent part of the majority of the diseases, right? You take like arthritis, psoriasis, lupus, and so on. But like these are like highly chronic disorders. Like, but like despite having very potent anti-inflammatory drugs and immunosuppressant drug, the overall therapeutic outcome from treatment for these patients are rather poor. The reason is like, if you see the level of disease in these patients, right? Like it's never like a steady state. It's highly fluctuated, right? Like because like you have an increased period of the disease, you have a steady state, occasional remissions, and the occasional flare-ups, and all it happens. And unfortunately, today we do not have a reliable, non-invasive method to measure the level of inflammation a patient has. So that makes it very difficult for the clinician to prescribe right dose of the drug in the right time, right? So therefore, if you want to solve this issue, like you need to have a material which can release the drug only when the disease there. When the disease is not there, shouldn't release the drug, right? But since we can't even measure the level of inflammation, it is very difficult to impart any such properties into any material, right? So therefore, it would be only possible if a disease can control and decide when to release the drug and how much to release the drug, we might able to solve this problem, right? So that's where like we have been developing this disease responsive drug delivery platform. How we do is like, first you need to identify a biomarker that is specifically produced during the disease condition. So this biomarker could be like an enzyme, peptide, cytokine, and so on. Okay. Now, like you develop a drug delivery system which can be, implanted in the body, but in the absence of biomarker, they shouldn't release the drug. But when it comes in the contact with the biomarker, that should able to trigger the release of the drug. And the released drug goes and elevate the disease and that stop producing the biomarker that stop uh, um, releasing the drug. In this way, like, disease itself can control when to release the drug and how much to release the drug, right? The key is like how you design this material, which will have such property, right? So for that, like we design a specifically this amphiphilic molecule. Uh, these are small molecules which have the ability to self-assemble to form this nanostructure. So when they undergo robust self-assembly, it can encapsulate drug and you can keep them as in the body as a depot. Depend upon the self-assembled strength, they don't release the drug in a non-specific manner. But these structures, you put it in such a way that like when it comes in contact with the biomarker, they're chemically able to degrade this material to release the drug in an on-demand manner. Okay, so that's the overall approach what we take. And I'll tell you like how this concept could be used for multiple medical applications. For example, like, this we have used to protect the transplantal organs from the rejection episode. As we know, like, when you post-transplantation, people have to take immunosuppressant throughout their life, right? Because like, our body try to reject them. But like, uh, the major issue is like, these people have to take immunosuppressants every single day, like throughout their life. And highly systemic, right? That leads to their entire immune system get compromised. It's a global immunosuppression. And that's why like this patient, like a highly vulnerable, a small infection could be pretty lethal for them. right? And also high systemic exposure can lead to severe other side effects. So what we have done is we have developed this immune responsive biomaterials, and we are particularly interested in hand transplantation. Post-transplantation, we can and take the known immunosuppressants and inject them as a small depot under the skin, and they sense the immune activation and release the drug only when it is needed. When immune system is not active, they don't release the drug, and this is also much more localized manner. Therefore, we have shown that if you do like a once in a few months one injection, that is enough to protect the transplanted organ till animal is alive without causing the global immunosuppression and without causing any non-specific organ toxicity. And we have shown that in the small animals like rats, and also in the pigs also we have seen fantastic efficacy. Right now we are just starting studies in the monkeys, and the post-monkey studies will be going for the human trials. And similarly, like the same concept, we have shown that it could be used for improving the therapeutic efficacy for the treatment of inflammatory arthritis. For example, as you know, arthritis primarily affect the joints, right? But like people have to take large doses systemic, and that leads to severe side effects. That's why people have started taking directly intra-articular injections in the locally, they can inject them. But the current material, despite of whether you have the inflammation or not, they release the drug few days to few weeks and disappear. But according to protocols, we can do only 2 injections per year. Otherwise, we end up damaging the tissue, right? And therefore, the inflammation responsive material, what we developed, we have shown that we can inject them into the joint. If there is no arthritis flare, they remain stable without releasing the drug, despite of your mechanical stress while walking and all, right? But whenever inflammation flare comes, that triggers the release drug, and it curtails the inflammation in the beginning itself. So because of that, we can significantly reduce the frequency of injections, have the much better therapeutic outcome. And similarly, we have shown that now we have added another layer of design, like not only inflammation responsive, but inflammation targeting material, where we can take this material which can go and sense the inflammation, because as you know, when you have Crohn's and colitis diseases, you have the patches of inflammation throughout your gut. But when you take your regular drug delivery systems, you get the drug release and you get the dose. A disease inflamed tissue get the same dose, and your neighbor healthy tissue also get the same dose. So that leads to severe side effects. That's how the overall therapeutic outcome is rather poor. So what we have shown is the material what we develop, which can go and sense this gut and bind to inflammation and specifically and release the drug only to the site of inflammation. So this I'll discuss a little more detail in the coming slides. So then with that, we could able to enhance the therapeutic efficacy significantly. So as I shown, this platform technology could be used for multiple medical applications. And to translate this technology, we are particularly interested in translating them into next level to the clinic, to the market. So for that, I formed a startup company in Boston, where we licensed our IP, and then through that, we have already completed phase 1 studies for one of the clinical indication. We're going to start phase 2 studies pretty soon. So with that, I thought I'll talk a little more about our prophylactic technology in detail. One of the technology what we are developing is to protect the farmers from pesticide-induced toxicity. Okay, typically, if you see all the pesticides, these are highly neurotoxins, right? They are designed to kill the pests. And what happens is you have this enzyme called acetylcholinesterase, which plays major role in all your neurological and neuromuscular functions by controlling the concentration of a neurotransmitter acetylcholine at the synapses and neuromuscular junctions, right? But all these pesticides, they are designed to go and covalently bind to this enzyme, irreversibly inhibit, so that when you have this quantitative inhibition of this enzyme, that lead to severe neurological toxicity and paralysis and death. That's how pesticides, when you spray on the pest, they die, right? Instantaneously. But unfortunately, these pesticides do not discriminate between pest and human, right? And because we have same enzyme, same function. So when we get exposed, we have the same toxicity. And typically, if we see farmers in India and developing countries, this is how they spray pesticides without having any protection. It's almost like a walking under the poisonous rain. It falls on their skin, they inhale, and they get severely exposed, right? Typically, like they get exposed to dermal and inhalation routes without, can we develop the technology to protect them? But like, before we start, we thought of like, see what kind of side effect they have. And typically, when we talk about it, like we'll go to conventional literature, but the conventional literature says that like, people have to go like, get exposed to like, years to decades, then, they start showing the chronic symptoms, which we couldn't comprehend at all because like, when they are such highly neurotoxins, how can't they have any acute effects? So instead of believing just on literature, what we have done is we launched a survey. We spent almost 8 months in the field and visited 60 villages. We spoke to like 200 farmers. It's not just by like, randomly speaking, like we used to time each farmer, like when they spray pesticides, spend a couple of days with them to understand what they spray, how much level they exposed and get exposed, what kind of side effects they feel, and like do they take any protective measures at all. I think that is one of the best decisions we have made because like that's completely changed the perspective on the pesticide toxicity because all these people, more than 99% of the people, like the same day they suffer with the toxicity, and like, depend on the dose they get exposed, they suffer with vomiting, diarrhea, eye fever, eye rashes, muscle pain, breathing disorders. If high doses exposure can lead to, blindness and death, right? And this happens every single season across the country. And that shows like, the kind of the unmet need it is and lack of the technology to protect them. So that's where we decided like, thought of go and develop the technology to protect them. So one of the first thought we thought is like, can we develop a dermal cream which can they apply on the skin, which can prevent the exposure, right? But like, it's just a physical barrier gel don't work because like, these compounds are designed to penetrate much faster. So just physical barrier, they don't work. That's why we wanted to have a chemical barrier gels, meaning like, you need to have a gel which can like, if any pesticide comes in contact, it should covalently attack, chemically react and degrade in such a way that this degraded compound can no longer inhibit the enzyme. Okay. So in that way, we can completely prevent the exposure. So, but if you see like typically like all the pesticides, right? Like you see large number of pesticides, so diverse structures, right? But all of them, like if one common chemical entity, what we see, these all of them are activated organophosphates, right? So the more labile they are, like more toxic they are. So therefore, what we thought is like, can we use this fundamental nucleophile mediated hydrolysis where you can convert this active uh, ester into an inactive acid, right? So then we can prevent the enzyme inhibition. So for that, again, like we need to have a really super nucleophile which can attack much faster upon under contact, right? So that's why we selected the class of compound called alpha nucleophiles. And because alpha nucleophiles are like a super nucleophile, their reactivity is super high. And there are several classes of alpha nucleophiles are known, but we selected oximes as one of the alpha nucleophile. I'll tell you the reason. And typically we can take this, a pyridine aldoxime based alpha nucleophile, which can connect to a polymer. Now, this oxime contained polymer can be by using appropriate your excipients, you can convert them into a gel, which can be applied on the skin. But as a control, we took like only a control with a polymer without having this chemical scissor and used as a physical barrier gel as a control. So once we have that, like, for example, you can go and take expose animals to three group of animals to this pesticide, either directly or through just physical gel or this oxime gel. And one way to measure the level of exposure is we can collect the blood or tissue before and after exposure and see the amount of active enzyme remains, right? The more inhibition happens, the more exposure. And as you can see, for example, if you take the blood, measure the active enzyme, And when you have direct exposure or a physical barrier exposure, you can see significant drop of enzyme activity compared to unexposed one, which is clearly suggesting that pesticide could able to go through the skin and inhibit the enzyme. But whereas when you have the polyoxime gel, you don't see that. And similarly, we have collected the tissue and we see the same. And we have done various other analysis about their neurotoxicity and so on, like this completely prevents them. So what we thought is like, okay, since this works, like, can we go and develop this for the human applications? And we went and developed the formulations for human as well. We have done safety studies and all, but like, we were still like, although technology works, like at the back of our mind, like, we're still thinking whether does it really solve the problem? Because like, there are two problems what we could foresee is like, one is the cost, because we're targeting the farmers that their purchase power is low. Like if it is cream, every time they have to keep applying, they have to keep rebuying, right? So which would be difficult. And another one is the compliance, because like, they have to apply all over the body before they spray. Not everybody might be comfortable. So, but again, like to decide by ourselves, we thought like we'll get the feedback from the farmers itself. So our neighbor institute, like Agriculture University, conducts this Krishi Mela where thousands of farmers come in within few days there every year. So we went and displayed our technology and got the entire feedback. They completely resonated like in the doubts what we had. So because not everybody is comfortable like applying, every time they spray. So then we thought like what could be the alternative technology, right? So typically if you see like, people like when they spray, some of them they believe that fabric can prevent them, so they wear full sleeves and they wrap around towels and all. But actually they don't protect, but rather it has negative impact the more if they get absorbed and remain as the reservoir and so on. So what we thought is, can we develop a fabric, okay, which is instead of a simple physical barrier fabric, which can again chemically deactivate the any pesticide that comes in contact, which can be wearable, right? So then we can solve this problem. So for that, again, we have designed another molecule like where like you have this alpha nucleophile, but it can be covalently attached to the fabric. Now this activated fabric, you can stitch into any form we want, like the coat and mask and so on. So once we have that, again, like we went and exposed to the animals, and again, like post three group of animals were exposed. One is like regular normal fabric without this oxygen functionalized one. Another one is oxygen fabric. And the post exposure, if we collect and see again a significant drop of enzyme activity when you expose directly or through normal fabric. But whereas when you have oxygen fabric, that is completely prevented. Same we see in every single tissue what we collected and show that is clearly suggests that indeed this oxygen fabric can break down the pesticide before it entering into the body. And similarly, like, but if you see farmers, like, they spray pesticide every single, they repeatedly, right? They keep getting exposed repeatedly. Wanted to see, can we prevent that repeated exposure? And for that, like, again, animals were exposed every single day, multiple doses, multiple days. But like on the same fabric, they were re-exposed to the pesticide. And if you measure their entire survival rate, if you see the animals repeatedly exposed either directly through normal fabric, all of them they die like within 4 days or so. But whereas the when you have the oxygen fabric, despite of like repeated dose exposure, it's a completely protects them 100% survival we see. So we're suggesting that indeed these fabric act as a catalyst rather than the stoichiometric. It can keep deactivating as long as they wear it. So but this is for the large doses exposure, right? But when you people even small dose exposure they can get, that can lead to severe neurological damage. Okay, so we wanted to see is there any small amount also can get through this and cause internal neuronal damage. So one way to measure the neuronal damage is to measure the sciatic nerve damage, because your sciatic nerve is something controls your limb movement. Okay, the way you measure is do the gait analysis. You can take the animal, put like a different color on the palms, and make them walk and like collect their footprints. And once you have these footprints, like you can measure the distance they take between the steps and also angle between their toes and so on. So all these numbers, there's a complicated equation you can put it in that, like, but what you do is like you measure something called sciatic functional index. Okay, so the thumb rule is like when you have completely healthy neuronal system, this number always come between like plus 11 to minus 11. That is the range where you see. But if anything number comes below minus 20, considered as a neurological damage. The more negative number is, the more neurological damage. And if you see like the animals that was exposed directly before, they were completely healthy, but like after exposure, single dose exposure, you can see a significant drop of this SI5 value. That means like severe neurological damage happened. Same way, when you get exposed through your normal fabric, also you can see a severe damage that occurs. But whereas the same dose of exposure when you do with oxime fabric, is completely protects them, which is clearly suggesting that even small amount of pesticide is not able to pass through um, this fabric to cause the neuronal damage. So another one is, as I mentioned, like when these pesticides inhibit this enzyme, and that causes the accumulation of neurotransmitter acetylcholine at the neuromuscular junctions, right? That leads to involuntary signaling. So we wanted to see whether we can prevent that. Like, for example, your NMJ signal you can measure by electromyogram. And this is a healthy person who never got exposed to the pesticide. And if you measure the signal, you see there is no neuronal signaling, right? Because, we're looking for the involuntary signal, right? Because this person is not moving any muscles. Now, if you do the same thing for a farmer who got exposed to the pesticide, you can see the neuromuscular junctions like highly activated, their neuronal firing. That means like, again, like this person also not moving the hand voluntarily, right? So this completely involuntary moving. That's why like you see like when people get exposed, they have this tremors, shivering and tremor. That's how it occurs. So we wanted to see, can we prevent that by using our oxime fabric? So again, like to do that, like we measured this electromyogram on the rats, and if you keep them under anesthesia, you can see like absolutely there is no signal. But when you get exposed to a single dose exposure, either directly or through normal fabric, you can see is a huge firing of neuromuscular junctions. So which is suggesting that indeed they got exposed and they inhibited the enzyme that lead to accumulation of your neurotransmitter that led to the signaling, right? The same dose exposure through oxime fabric, again, you can see a completely prevention of such phenomena, which is clearly suggesting that indeed this material has the ability to break down before it entering pesticides entering into the body. So these all are for the dermal application, right? The body. So we wanted to see inhalation also, can it be prevented? So for that, like, we build this box where we can fill this box with the aerosol of the pesticide. Now these animals breathe these pesticide aerosols either directly or through a mask made by normal fabric or oxime fabric. And the post-exposure, again, you collect the blood and tissue and see how much enzyme inhibition happen. As you can see, the animals that breathe this pesticide aerosol either directly through normal fabric had a significant drop of enzyme activity, but whereas through oxime fabric is completely prevented, which is clearly suggesting that indeed this material has the ability to break down even through nasal route as well. So once we have that, again, like as I mentioned, like we wanted to translate this technology to do that. So like we formed a startup company in Bangalore, SAP Health Private Limited. Then we developed this suit called Kisan Kavach, and this is how it looks. And this has both pant and shirt with a hoodie and the inbuilt mask and so on. Now this suit has been like distributed to multiple places and farmers in various states, and we're getting like a phenomenal feedback from the farmers. People like were instantaneously used to feel entire body pain. They were able to get relief instantaneously. So I hope I shown that like how our Kisan Kavash, especially anti-pesticide fabric, went from the lab to the field, where right now people are able to use farmers in several states to get the protection from this completely pesticide-induced toxicity and so on. So with that, I would like to quickly go through like a couple of a few other examples, like, we were also like interested in developing the novel blood bags to enhance the quality and shelf life of stored blood. Uh, so typical, as you know, like a healthy blood transfusion is a life-saving process, right? Like, typically in the India alone, if you take like every year, we require almost 12 million units of the blood for the transfusion. But whereas from the donations, various blood banks and, blood donation camps, we get like a less than 10 million units of the blood. But despite of that scarcity, like, every year, almost 5 lakh liters of the blood is discarded because the quality declines during the storage, right? So that's why there is a huge need to increase the quality of the shelf quality and the shelf life of the stored blood. But people have been trying that, right? So for example, like what happens is when you collect the blood, it's up to 42 days you can store them to transfuse. But however, only up to 21 days stored blood is safe to transfuse, because after that, the quality of the blood declines. And if you transfuse this old stored blood, that can lead to severe systemic immune activation and internal organ damage and so on. So the reason why it happens is when you have this stored the blood, they start producing something called DAMPs. These are like, extracellular DNA, nucleosomes, polyunsaturated fatty acids, hemes, and proteins, and so on. So all of them, they attack RBCs and causes the oxidative stress, right? So because of that, the entire quality goes down. But if we see, like so far, all the technologies past couple of decades, they have been trying to develop a more preservative solution. These are nothing but different buffers to reduce the oxidative stress on the RBCs. Although they work moderately well, but like, one of the major problem is like, you do not, they are not addressing actually cause of the problem, right? Like, because your damage causing agents are still present there. So it's always war between dams and the this protection from the buffers. Therefore, what we thought is like, can we, if you remove these dams or damage causing agents from as they form from the bag, will are we able to going to develop the or increase the shelf life or the quality of this blood? So, but the question is like, these are like a very diverse structures what you have, right? Like you have, DNA, histones, nucleosomes, proteins, and lipids, and so on. So, how one should design a material which can scavenge all of them, right? So, but if you look from the chemistry point of view, they are like, a highly charged molecule. They are either negative or positive charge. So what we thought is if you design an appropriate a polymer material which have the complementary charges and also like, the chelating or binding ability, intercalating agents can be able to pull them up or scavenge them. So for that, like we have systematically designed and optimized these polymers, like with the complementary charges, and we can convert them into this nanofibrous scaffolds like a bag. And now we can take this, and if you take the dams containing blood and pass through this bag for like keep them on like a 10 to 15 minutes and take out, and it captures all the dams and releases the healthy blood cells, right? So I quickly summarize our overall requirement. What we have shown is like, when when we treat this compound, now use this technology, even 42 days old stored blood quality is as good as freshly collected blood. Okay, that much quality we can increase. Why I would say is like, the quality is defined by the transfusion efficacy, right? Uh, for example, according to FDA guidelines, when you transfuse the blood, the 75% of the transfused RBC should be in the circulation after 24 hours, right? Then only it's a qualifies as a successful transfusion. Because if it goes down, then, people have to take a repeated transfusion. So, for example, you take the freshly collected blood, okay, you transfuse them, and after transfuse them, you collect the blood like, at different time points and measure how much the transfused RBCs were in circulation. For example, if you see, not at 24 hours, you can see there are 80-90% of them are in circulation, but not only 24 hours, even 48 and 72 hours also, you see higher number in the circulation, right? Because you freshly collected and transfused them because there is no damage. But now if you do this for old stored blood. Oh, by the way, the stored blood we have done with the mice because human trials we can't do immediately. So for the human, the mice blood, like 14 days equivalent to 42 days. That is like a 1 day mice stored blood is equivalent to 3 days stored of human blood. Okay, so now 14 days stored blood, if you transfuse them, if you see the post-transfusion 24 hours, again, they are like a required amount in circulation, about 80-85%. But you can see 48 hours, less than 50% it fell down. That means it's already very fragile. The quality has declined significantly, very fragile RBCs. That's why people have to repeatedly, they need to keep getting the transfusion. But now, for this, when we use our technology, instead of storing 14 days continuously, in between, once you pass through these scaffolds and store them back and 14 days, and now you take this 14 days stored blood and do the transfusion. And as you can see, not only 24 hours, even 72 hours, you see they're almost 80-90% in the circulation. That is as good as freshly collected blood. That means by removing dams intermediately, completely prevented the damage of the RBCs, so that it could enhance the storage of the quality of the blood. So again, to translate this technology, we formed a startup company. This is just very new startup company. We are going to start the manufacturing the blood bags, and then subsequently we have to go and do the human trials, blood transfusion trials, and so on. So with that, I'll give one example, another the preventive technology, where we have developed this germicidal fabric technology to prevent viral or microbial infections. This is particularly when, when pandemic has started, wearing mask has become inevitable, right? But if you take all kind of masks, N95, surgical cloth, these are fantastic filtration devices, right? So that's how they prevent all the bacteria and virus getting exposed. But the clinical studies shown that the bacteria and virus can accumulate on the surface of this fabric and remain active for multiple days, depend upon the strain you have. So that means when you are trying to adjust and all, one can touch and potentially can get exposed, or if you just discard them, it can spread, right? So that's where we thought can we develop a novel germicidal fabric which doesn't allow you to accumulate on the surface of the fabric. But first, we wanted to quantify how much bacteria accumulation actually happens. So, for example, this is a typical agar plate. When you don't have the live bacteria, this is how clean it looks. But when you have the live bacteria, this is how you see the murkier plates, right? Now we asked people to wear 6 hours this mask, and after that we just took the swab on this mask and you look for the live bacteria. You can see that all of them harbors your live bacteria. The cloth ones even worse, they harbors much higher number. So which is clearly suggesting that indeed you carry live bacteria, they're accumulated on the fabric. So now we have designed small molecules, these are germicidal molecules. These are like a tentacle. They can go and interact with the membrane of virus and bacteria, and physically they rupture them and deactivate them instantaneously. In that way, when you made your mask with this germicidal fabric molecules, now they don't not only acting as a physical barrier, but it will deactivate any bacteria virus comes in contact. And we have shown that now for that, what we have done is we made the mask with this fabric, and we asked people to wear them not only 6 hours, but even 18 hours. And after that, you collect the swab and look for the live bacteria. You don't see any live bacteria present at all. And this we have tested with like both gram-negative, gram-positive bacteria, and also wide range of viruses, including SARS-CoV-2 and all. So again, to translate this, we formed a startup company in Tirupur called Color Threads. Then we manufactured the mask, but we wanted to quickly go to the public, and that's why we wanted to have a distribution partner. So we partnered with Van Heusen company, which is the Aditya Birla company, and through Van Heusen we launched under G99 antiviral mask. Not only mask, like we also launched a variety of apparels, because these are highly washable, reusable material. So in fact, this is one of the quickest translated technology, right from the concept to commercialization, launching into the market took less than 9 months. That is developing technology and also forming the company, scaling up, and manufacturing and launching them. So with that, I thought quickly take one example of our example in our drug discovery program. Particularly, we were interested in developing small molecular drugs for IBDs. But typically, if you think about the drug discovery, there are two major approaches people follow, right? One is natural product based, like you find a medicinal plant and identify an active drug component and use them as a drug. And other one is classical medicinal chemistry approach, where you do find a target and do the screening of the libraries and find the hit, and then subsequently develop a lead molecule and so on, which is like typical pharma follows, which is also cumbersome, like a heavy investment demanding one. So, but apart from these two, there's a new concept being evolved, very new concept is biomimicry based approach, meaning if we take our body itself, it's a chemical factory, they develop variety of molecules. So, and they do have this certain biological function if you understand them, but they don't act like a drug, but they can modulate the biological function. So if you understand their biological function and clubbing that with the chemical infrastructure information, one might able to develop the drugs much more efficiently. So that's the approach we wanted to take. And as I mentioned, IBDs, they comes in two variants, on Crohn's and colitis. Typically, if you see the etiology, right, when you have healthy condition, we have a single layer of epithelial cells, which are kept intact by these tight junctional proteins, like a molecular sutures. And this layer prevents any bacteria, commensal bacteria, getting exposed. That's how you have the homeostasis. But in this disease conditions, what happens? They lose these tight junctional proteins. It becomes a leaky barrier. That's how, your commensal bacteria keep getting exposed to the system, and your immune system start attacking. That causes infection, and that leads to inflammation and so on. It's like a vicious circle, right? So, but today, if you take all the drugs that are available, they are either anti-inflammatory drugs or immunosuppressant drugs. That meaning they are rather disease managing drugs, not disease modifying drugs, right? So the symptoms they can take care, but they don't really cure. So we thought there's a huge scope to develop the drugs to potentially cure them, right? So, but where to start, right? Since it is IBD, the gut. So we thought of looking at the microbiome there, because as microbiome is a double-edged sword, right? While it can provide beneficial effect when it's in homeostatic conditions, but in the dysbiosis can cause pathological conditions. And also, recently, people have shown that your microbiome also plays a role. It can control the function of your pivotal organs, lung, liver, brain, and so on. The question is how a bug that is sitting in the gut can control the organs remotely, right? How they do is depending on the diet what we take, these microbes convert them into active metabolites, right? And these metabolites act like a chemical cues to do their biological function. So one of the example is, for example, when people take the phytochemical rich diet, they are known to have the beneficial effects, right? Like cherries, berries, and pomegranates, and so on. what happens is that this phytochemical rich diet have this polyphenolic compounds, lecitins, and our bacteria, it can break down into polyphenolic metabolites called erythrins. Okay, and people have shown that one of the metabolites, such as like erythrin A, has uh, anti-inflammatory properties. Maybe that could be the reason why when people drink this juice or this phytochemical rich diet, they have the beneficial effect. But the issue is like, when people try to take this molecule and give them, and all these anti-inflammatory studies are done in vitro, right? But when you try to replicate in the in vivo studies, nothing worked, right? So then we thought maybe that is a starting point we can start with. So if you look at this closely, this structure, right? Like, this compound, it's basically a bisphenol, like, but it has a lactone, a cyclic ester, keeps them this compound in a planar manner, right? So, but like when you orally you take them, it can completely degrade it, break down due to your low pH and your digestive enzymes. But once it breaks down, it becomes a propeller structure. No longer it has the planar structure. That's why it loses the complete activity. It can no longer bind to its target. Therefore, first of all, we wanted to do this two-pronged approach we want to take. One is like, can we take this and develop a delivery system where you can protect from the degradation in your gastric solutions and go and specifically release the site of inflammation so that we can have the beneficial effects. So, but as I mentioned, like you have these patches of inflammation, right? You need to have a system which can go and specifically bind to only where you have the inflammation, like a glue. So if you release the drugs only there, then all the drugs can be go to only inflamed site. Your healthy tissue can remain unexposed. So, but the question is like, how one can impart such specific adherence property, right? As I mentioned, like typically when you have the inflammation, you have the leaky barrier, right? This causes the infection. So to curb that, our immune system start producing these high concentrations of this cationic proteins and antibacterial peptides to prevent the infection. But all of them, if you see, they are highly cationic charged, right? If you see that your inflamed tissue like a cationic charged bed compared to your neighbor healthy tissue, either neutral or slightly negative. So what we thought is, can we use particles with the appropriate complementary charge, which can go and specifically bind to the proteins that are expressed at the site of inflammation. So that was the approach. So for that, again, we took like a various excipients. We made the solid core where we can encapsulate these drugs. And we also use this typical polymers, which can provide the appropriate charge density, negative charge density on the particles. And we have right size and the right density of the charge density on them. And again, we wanted to see whether it can protect from the gastric fluids, gastric and intestinal fluid. You can see you don't see any burst release from these particles and so on. So that protect them. Then we wanted to see whether it can bind to the proteins that overexpress at the site of inflammation. The transferrin is one of the protein expressed at the inflammation site. So that was coated with the glass plates, and also mucin was exposed in a healthy mucus, right? That also coated. And you can take this fluorescently labeled particle and incubate them and see how much particle they bind. No surprise here, like because all the particle, because you have the negative charge and positive charge, so they nicely bind them, right? Um, so the question is, can we get the similar level of binding in vivo conditions, right? So for that, like we can take the animals and you have healthy colon, right? Like if you give these animals dextran sodium sulfate for 7 days, they develop this disease and you have now inflamed colon. Now we can take the particles and give oral gavage them, and after 24 hours, we can collect the colon and see how much particles they remain bound to this colon, right? And when you do that, like, when you expose to healthy animals, you don't see any fluorescent signal at all. That means like, the particles couldn't stay there because it's after 24 hours, right? They might have completely excreted out. But whereas when you have this colitis animal colons, you can see the patches of inflammation, patches of this fluorescent signal. That is, you know, That means this particle were able to specifically bind like a glue and remain there. Despite of all your bowel movement and all, right? Like, still the particles are able to bind there. So which is clearly suggesting they have the ability to bind them. But now we wanted to see, although this, again, it's a murine system, right? Like, can we able to get the similar binding efficacy human? Okay, so for that, like what we have done is, again, we took the uh, biopsies from the patients who suffer from the colitis. From the same patient, we took the tissue where they have the ulcerated tissue, inflamed tissue, but the same person, we also took the healthy tissue and ex vivo incubated with particles and washed them to see how much they bind it. Okay, and as you can see, this is a healthy, when you have this, all this five, in a healthy tissue, you don't see any significant amount of binding, but when same people, like when you collect it from the ulcerated tissue, you can see the significant binding, right? Even individual manner, if you measure, like, for example, this person, healthy tissue have certain amount of binding. The same person, when have the inflamed tissue, it binds much higher. So which is clearly suggesting that indeed these uh, uh, particle has the ability to bind them, right? Now, once we have them, then we went and tested their ability to have the therapeutic benefit, right? So for that, again, like we created, give the animals DSS, which create the disease. Now you give the drug, either directly or same dose you give by encapsulating it in nanoparticle and see. And then what we see is like when you provide them through nanoparticle, you can see a complete protection. One way to measure the therapeutic outcome is the colon length, because when you have a healthy colon, when you have the disease, they lose the colon because of inflammation and all, they shortens it up, right? So when you give just drug, you don't see any protection from the loss of colon, but when you have the with the nanoparticle encapsulated, you can see huge beneficial effect, which clearly shows your histology also, because when you have through nanoparticle when develop, you can see remain of the your cryptobullar structure, they are intact, whereas when you have the disease, they completely lose it, right? So then we wanted to see how robust it is. So what we have done is like after fully developed disease, we gave one single dose of this nanoparticle. Even one single dose is completely protected them from the colon loss, which is like a super good, right? Like, once, although one side, like we are super excited that like, this is almost like a reversing the phenotype, but we are also skeptical because this compound is supposed to be an anti-inflammatory agent, right? And that means a disease managing drug, it's supposed to be. But with this, when you have the disease managing drugs, with a single dose, you cannot have such phenotype, right? So uh, that's why, but as I mentioned, healthy conditions, you have this barrier, healthy barrier, right? But the disease conditions, you have this leaky barrier. But as long as leaky barrier is there, just giving your anti-inflammatory drugs wouldn't have much impact. With a single dose, definitely not. That means probably this compound is doing far more than what it is just an anti-inflammatory agent. Our hypothesis was probably this is helping to restore the barrier function. That means it's cutting the source for the infection. That's how we are able to recovery much faster, much better. So to do that, we went and tested this hypothesis. And also like what we have done is like we also wanted to make a synthetic analogs to see more stable analog we made, like basically converted your lactone into cyclic ether, which cannot be degraded by your low pH or your digestive enzymes. Now we went and took these compounds and exposed to colon cells. When you treat the colon cells, you can and measure the tight junctional proteins, because one way to restore the barrier function is having the overexpression of tight junction proteins, right? As you can see, like all of them, they upon treatment with this, they overexpress tight junction proteins. Um, and also then we wanted to see the barrier function, can it be repaired? And for that, like in vitro barrier function was created by using transfer plate. When you have the monolayer cells, you can, on top of it, like you can put this Swiss Dexon, a polymer, and see how much it passes through, right? For example, in healthy conditions, you have certain amount of polymer diffusion. But when you have the LPS, if you give the LPS, it breaks the barrier. Okay, then you see the increasing the barrier diffusion, but upon treatment with these compounds, is completely restores. That means it is repairing the barrier. That's why it shows its efficacy. Then we went and tested its ability for the in vivo as well, right? So in vivo to measure is like uh, what you do is like you do the fixed exon you can give them orally and collect serum and see how much this huge polymer comes into the circulation, because in a healthy systems, like since it is a heavy polymer, large polymer, it doesn't come much, but when you have the disease, you can see like a huge amount of polymer comes diffuses into the circulation, which shows the leaky barrier. But when you have treated with these compounds, it's completely restored the barrier function. And also like it has anti-inflammatory, and you have through histology also it shows the beneficial effect. And not only in acute, but even in the chronic system also, we have shown that like it can completely restore the barrier function. So that means your gut is getting repaired. That's why you see the curing phenotype. And again, we collected the tissue and measured the in vivo also tight junction proteins were exposed. And then after that, like then we our hypothesis is like, upon treating with this compound, it is overexpressed, stimulating the gut cells, epithelial cells, and overexpressing the tight junctional proteins, that is restoring the barrier functions to have the beneficial effect. So then we wanted to see the mechanism. How does that work? Then we did like a series of RNA-seq studies and all. Then the bottom line, what we found out is these compounds go through AHR and RF2 pathway to have this beneficial effect. So to test that, what we have done is like we repeated this experiment with the AHR knockout. If it is wild type, as you can see, they have completely protected. But when you have the AHR knockout, if you see, they don't see any beneficial effect. That means like, it couldn't able to restore the barrier function. It has the leaky barrier, and also they couldn't able to protect from the loss of colon function. Similarly, NRF2 knockout mice also, we see the same effect. Absolutely, they don't work. So which is clearly suggesting that indeed these molecules are go through AHR and NRF2 axis to have the beneficial effect. And then we have shown this efficacy in multiple models, like, chemical induced models, spontaneous models, and so on. And to summarize, like, this molecule are able to repair the gut barrier, leaky barrier, and restore the barrier functions to have the beneficial effects in IBDs. Again, to translate this technology, we formed a startup company in Boston. It's a pharma company. Because your first line of compound, they are like just models, but they don't really act as a drug. Precise is know this very well. Just activity is not, efficiency is not everything, right? This is just one part of the drug like property, but you have better for other safety and studies and all will be there. So these compounds are like a super genotoxic, and also that's why like now we have done extensive medchem around this once we understood the basic biology of this. Now we have 4 molecules which have like a phenomenal efficacy and also high safety profile. Now we are working with the FDA for going for the phase 1 trials pretty soon. So with that, I would like to summarize, like I've shown that, like, in our therapeutic technologies, we have shown that how the disease responsive biomaterial could be used for, protecting transplanted organs and improving the therapeutic efficacy of the arthritis and also in IBDs. And the prophylactic technology also has shown that, like, how we could able to develop the technology to prevent the pesticide exposure for the farmers and also increase the quality of the stored blood. And in the drug discovery, could able to develop small molecular drugs, which could be very efficient for disease modifying drugs for the treatment of IBDs. And as I mentioned again, like we are equally interested in the translate these technologies, and various startups were formed based on these technologies, and they are in the various stages. Some of them are into phase 2 clinical trials, and a couple of them have already launched in the market, and so on. With that, I would like to stop here, and I would like to thank all the people, like the fantastic postdocs, graduate students, masters and interns who developed this better of technologies. And this is my current lab, and I would like to thank all our collaborators and also various funding bodies for their generous funding. I thank you all for your kind time, and I'm happy to take any questions if you may have. Thank you.
Yeah, thank you, Professor Vemula. I have thoroughly enjoyed your talk. I hope it is the same with our audience as well. Yeah, I see there are questions. Certainly, there would be, since the talk was very nice and interesting. I'll take those questions one by one. Yeah, so first is Dr. Shubham Sarkar. He's asking pralidoxim, which is an oxime that is already used therapeutically in emergency setup in IV route, along with atropine for organophosphate poisoning. It is common clinical practice in ED for organophosphate poisoning. So in your case, you change the route of administration and also using it prophylactically before the exposure. So is it correct?
Yeah, so this is a completely different approach. That was the post-exposure, and also in vivo treatment doesn't deactivate the peptides. That is to reactivate the enzyme. Okay, but which has a huge limitations. In fact, like last time any drug for the treatment for this pesticide poisonings was in 70 years back. Proloxam and all came that long. Uh, but the problem is like they work only like if you bring the pesticide poison patients within like an hour or so, hour to couple of hours. Then only these drug would work. Anything beyond that, your enzyme get aged and doesn't work at all. So that's a completely different approach what we are
Okay. The next question is from Sudeep. He asked, the delivery system of controlled release is fascinating. Could you please explain more about the kind of lipids which were used or discovered? And are these standard FDA approved excipients? Follow-up question. Do you also optimize the loading of API? And are these pH sensitive? That would be helpful for GERD, acid reflux.
Yeah, so to the answer with that excipients, yeah, generally we take these excipients from grass agents, generally because they're safe agents, to just to have any, not to have any surprise toxicity and all. But however, as you know that no matter how you can take the excipients, but when you make the formulations, together formulation will have a different properties. So one need to do extensive safety studies for them as well. So, but they are optimized in such a way that with the current formulation, they are completely biocompatible. They don't have any side effects yet. And some of these MTD and all goes to 2 grams per day for even a month dose. So they're pretty safe. And for the second question, they are not pH sensitive. In fact, they are designed to be not pH sensitive because otherwise in stomach and all they can get degraded. So if but if it's needed to develop pH sensitive, one can develop. Okay, the current system is not, is depend upon. That's why typically we, at least our approach, we don't like to have one system works everywhere and you can try to apply, but rather you find the problem and custom design exactly what fits for that particular problem and develop the system, optimize system in that way.
Yeah, I'll take next question. It is from Sudeep itself. The packaging industry, they're trying to solve the barrier issue and develop sustainable packaging. So are you working in the similar space?
Packaging industry. Sorry, can you repeat this is regarding what, like barrier issue? I didn't understand what barrier.
I'll just
Yeah,
rephrase.
maybe if they can little more specific because I
Yeah,
couldn't understand the question. I'm
yeah,
just reading also, but
yeah, yeah. We can take then. The question from Cheryl Parakil. Though 3D printing of protein receptor is not well established, I wonder if such a technology could be used to integrate with the amphipathic assemblies to sense disease biomarkers.
Yeah, it's technical, it is feasible, but again, we need to see what would be the advantage. You see, if you are looking for any specific shape, it's a 3D printing, it gives the advantages, any given shape you want, we can print them, right? So whether in the current applications, we don't need them. All you need, like, subcutaneous, you can inject them a small depot, or in the joint, you can generate depend. So in that way, like, we really don't need it. But the question is, if you needed to have any shape specific, then 3D printing would be highly beneficial. Um, Again, like the protein receptors, I am not sure. Again, protein receptors is again amphiphilic assemblies. They're completely different. These are like bottom up approach. Okay, depend upon the how you modulate your chemical structure, that defines the strength of your self assembly. The kind of nanomaterial they self assemble to form will change based on the chemical structure. So that's also good and bad. Like, it gives a lot more opportunity as to specifically engineer the one what we wanted to have. So I'm I'm not sure whether that answered the question, or like
Yeah, yeah, maybe we can take the next question from her itself. Yeah, phytochemicals in fruits are effective as they're present in fiber matrix, which is broken down and beneficial to the intestine. Is it possible to mimic this fibrous matrix system in delivering the phytochemical of interest in treating IBD?
well, you see, for example, if you take we take the current example itself, right? We take the erythrins. Okay, if you take pomegranate diet, like it can have the beneficial effect, right? But there are a couple of huge issues. A, uh, recently only people have identified exactly which bacteria actually breaks down to form this active metabolite. Okay, and 40% of the population across the world don't have this bacteria, right? So then no matter how much we take, it doesn't work. Okay. And the second, say you have it. So even if you have it, the this compound is very not very efficient. Subtly it works. In order to have meaningful doses, to have like a therapeutic benefit, okay, people have to take every day like 5-6 kilograms or more of this diet, which is practically not possible, right? So that's how, like, that's why what we see is like, instead of taking them as a drugs, but if you uncover the actual the biology how they work, and by combining that information, one can develop molecules much more potent and much more efficient. Yeah.
Okay. Next question is from Shradeep. I think this is from the last part of your talk. Um, he says, "How are nanoparticles helping? Is it because of slow release?"
Yeah, so nanoparticle particularly, they are helping to increase the residence time at the site of inflammation, and it's almost like a local delivery. You see, on a hand, I can, like, do direct injection as a local delivery, but gut I can't give that, right? So that's why the nanoparticle, these design nanoparticle, when you take them, and instead of going and binding everywhere, what they do, they sense and they bind to only where you have the inflammation, and sits there and release slowly. They release the drug. That means like you're delivering the drugs only at the site of inflammation. Your healthy tissue completely you can minimize. So that's how nanoparticles are working.
Mhm. Next question is from Seema. I think this is regarding the synthetic analogs. Any off-target effects due to its negative charge? Yeah, assume it's about the analog.
Yeah, the particles. Yeah.
Yeah, yeah.
So we haven't seen anything yet, Because one thing what we have seen is like a good part is like if there is no inflammation and all right, they don't remain that they excreted out. Okay, they don't get accumulated. So if they have long-term accumulation, you might have side effects. You don't know. But like what we have seen, like they cleared from the body. They excreted out much faster. So that's why even dose one can take much higher because it will go and stick only if the inflammation is there. Otherwise, they will be excreted out. In that way, like it will
Mhm. Yeah, another question from her. Is there a categorization for your G99 plus mask, such as biosafety levels?
I'm not sure the biosafety levels. How does that matter here?
Mhm.
Because this is used as a barrier, okay? But the barrier, but which also deactivate any pesticide. Any bugs comes in contact.
Mhm.
So, and we have shown this with the bacteria, virus, and fungi. Okay.
Mhm.
So I am not sure biosafety level. Biosafety level doesn't come from the mask, but rather the biosafety levels are decided with the kind of bugs we get encountered, right? You know, either viruses or bacteria, what kind of bacteria we get encountered.
Then next question: could this inflammation sensing technology be adopted for the treatment of difficult to treat diseases such as lymphatic filariasis, elephant foot, or even COPD?
Okay, COPD, yes, we can. Okay. In fact, we're thinking of starting a program on that. Other one, Elven food, I am not aware of the, I don't know much details about this disease, so I can't comment on that. But COPD, yes, yeah, we are going to do that. Yeah.
Okay. So now I'll take the opportunity to ask a couple of questions that I had. So regarding this oxime fabric, so is it sensitive to, let's say, acid or alkali, let's say acid hydrolysis, during the acid rain or something like that?
No, it's not. That has
Okay.
been optimized in that way. Yeah.
Okay. And then does your QAS molecules work against all kinds of cell membranes? Let's say rupturing all kinds of membrane, because there are some bacteria or some algal things. They modify their cell membranes. Let's say peptidoglycan layers, and they add. There's a much complex bilayer that they do have. So is it affecting against that as well?
So right now, what we have shown is like they work for like both gram-negative, gram-positive bacteria.
Mm-hmm. Mm-hmm.
And of course, their membrane is quite different, right? But still, they do work.
Mm-hmm.
And again, like we tested for all usual suspects, escape pathogens and all, they work against all of them.
Mm-hmm.
Um, so again, like it's also there's a lot of optimization went like not like one single molecule works against all of them. So
Yeah.
that's where a lot of optimization went through to find that one molecule which can work against all of them. But it's
Mm.
also the speed also important, right? How quickly it will deactivate as well. Yeah.
Yeah, so that's all from my side. So yeah,
Yeah, just to
thanks.
say that your Alzheimer one, what you have asked,
Uh-huh.
right? There are a lot many other parameters also we optimize. For example,
Mm-hmm.
like mm, if you see there are go-to format, there's pesticides are a lot more unorganized market. Also, you see hundreds of them, like
Yeah.
and you need to have one suit for all of them because if you make like multiple, you're targeting the farmers. Most of the time, they can't even read the labels. If you tell that you use this particular class of pesticide, you have to use this suit, then it beats the purpose. So that's why like we systematically optimize that, like having one system which would deactivate all these
Mm-hmm.
classes that are commercially available today. And also it's highly washable because it has to be, if it is just disposable, it doesn't work. So that's why
Mm-hmm.
it's a completely washable, reusable fabric. This suit like people can use easily a couple of years. For example, even when 150 washers, it still retains its activity. Basically, as long as fabric is there, it should work. And also, but when you know that, like when you have oxime kind of nucleophiles, often they become very sensitive to UV light, right?
Yeah.
They lose the activity. And farmers, they work under only sunlight.
Sun. Yeah.
So that's how structures are optimized in such a way that, like, under sunlight exposure doesn't lose this activity. And also that's how and also like we are doing the chemical reaction basically on the surface of the fabric, right? And it's a temperature dependent, but it should work like, peak winters and, hot summers and cold winters, it should work. Same efficiency. So that's how, again, the molecules were designed in such a way that it works in all seasons. So that's how like a lot of optimization went to come to the final. Yeah.
Yeah. Great. Again, thank you very much for the wonderful talk. And now I'll ask Dr. Seema to give a word of thanks. Seema.
Thank you so much, Dr. Vemula. I would really like to extend our heartfelt gratitude to you for agreeing to come and present at our Fusion Talk series. And I can honestly say that this is just an astounding body of work that you presented today. I think I'm hearing you after a gap of a decade, and it's just amazing. We are particularly grateful for your willingness to share your journey and the fundamental technologies that you have so successfully translated, for the benefit of the society. So it was really our pleasure to have hosted you today. Thank you so much for finding the time to come and talk to us about this phenomenally amazing work that you've been contributing to the field of science and technology and societal benefit. So I would like to thank you once again, and we look forward to your continued success, and hopefully any future collaborations that could potentially arise from this talk between your research group and perhaps with Prescience. So thank you once again, and thank you everybody for joining in today's event.
Thanks for those kind words, and thanks for the invitation. I think it's great to share some of our work and had a fantastic discussion. As you said, looking forward for future interactions and
Awesome.
see any potential we can work together. That would be
Yes.
great.
Absolutely, it'll be our pleasure and privilege. So thank you.
Thank you. Good evening, everyone. Take care.
Yeah. Thank you, everyone. Have a nice evening. Bye-bye.
Bye.